Here we see a micrograph taken from cardiac tissue, highlighting the cardiomyocytes. These cardiac muscle cells are normally 15 micrometers in diameter and 80 to 100 micrometers in length. These cells usually possess one, but sometimes two centrally located nuclei, as you can see on your screen. For the most part, cardiomyocytes are branched, and here we see a micrograph highlighting exactly that. Such branched cardiac muscle cells often bind to the cells in adjacent muscle fibres. It should be noted that these branched cardiomyocytes are more often seen in samples taken from patients with hypertrophic cardiomyopathy or enlarged heart muscle. Here we see a heart again. If we extract a muscle fibre out of it, we'll see that these are composed of myofibrils, and these, when observed even closer, are made up of sarcomeres. Sarcomeres, which are the functional contractile units of myofibrils found within the cardiac muscle cells. The sarcomeres contain actin and myosin myofilaments that are contractile proteins present in all muscle. The repeating units of sarcomeres along myofibrils are what give the cardiac muscle its striated appearance, a visual feature of altering light and dark striations observed under a light microscope. Here you can see these striations highlighted in green. As the contractile proteins in skeletal muscle are also arranged into sarcomeres, skeletal muscle also has a striated appearance. On the other hand, in smooth muscle, the contractile proteins are not arranged into such units, therefore giving it a smooth or non-striated appearance. Next, we can see the cardiomyocyte nuclei highlighted in green. As you can see, and as we have already mentioned, the nuclei are centrally located in the cardiomyocyte cells. This is one feature that distinguishes cardiac muscle cells from skeletal muscle cells, which are typically multinucleated. Cardiac muscle fibres possess transverse lines that stain dark during H&E staining, which are known as intercalated discs, and are visible under a light microscope. These discs represent the highly specialised attachments between adjacent cardiac muscle cells. Intercalated discs contain structures called gap junctions, which form channels between the adjacent cardiomyocytes. These junctions serve as communicating channels between the cells that allow for ions to pass between them, ensuring ionic continuity. This ionic continuity allows cardiac muscle cells to act as a single functional organ, or as a syncytium. Intercalated discs also contain desmosomes, which are intercellular junctions that form strong adhesive bonds between the cells. They function to bind or adhere the cardiac muscle cells to one another, preventing them from pulling apart as a result of repeated contractions. So it's worth remembering that intercalated discs and their desmosomes have an important role of providing mechanical strength and stability to the cardiac muscle, as to maintain its integrity when it's subjected to mechanical stress. It's also worth mentioning that cardiac muscle consists of bundles of cardiomyocytes such as the ones seen here in this micrograph. These bundles are surrounded by a tissue called perimysium. So far we've seen that cardiac muscle is made up of cells called cardiomyocytes. We've also seen that this type of muscle has a striated appearance owing to the presence of contractile units known as sarcomeres. It's important to remember that cardiac muscle is an involuntary muscle that is found in the walls of the heart. It's an involuntary muscle because it's regulated by the autonomic nervous system and is not under conscious control.
If you found this quick anatomy or physiology video helpful, you'll enjoy our video tutorials even more. Click on the button and you'll see what I mean. We have hundreds of videos available to our premium members, not to mention all the fun quizzes, complete articles, and atlas sections to solidify your knowledge. Click on the button in the middle to watch the full-length video and master anatomy and physiology.